So thank you for staying with us while we settle in. This is a different format, and it's my privilege to get to know this person better.
I think I'm mic'd up as well.
Right.
Hi everyone, I'm Sigma Mostafa. I'm Chief Scientific and Technology Officer at KBI Biopharma. We are an end-to-end CDMO provider.
Right, so celebrating 30 years, I see. As you reflect on this milestone, how has the mission changed? I see that patience and science is core to the mission, but over 30 years, I'm sure many things have changed.
So I'll start with what has remained unchanged. So our unwavering focus on getting products from cell line stage to all the way to clinic has remained unchanged. We have stayed focused on the scientific rigor as well as in terms of being a true partner for our clients so that it's not just a transactional relationship, but more of a strategic relationship with our partners. But over these 30 years, which is a major milestone for us, we were a technology company. Initially, we had particular centrifuge technology. We were also analytical services company for the first decade. And I joined in 2010, actually, and we became a PD manufacturing services organization. So I built the organization grounds up. We have 6 sites now and 4 commercial products and a big pipeline. So a lot has changed in terms of expansion, but our focus on how to get these molecules faster to clinic to prove their unique values has remained unchanged.
Yeah, so sort of frictionless drug development. COVID produced sort of the Amazonifying of our industry.
That's right, exactly, industrializing the process.
So before we go to logistical supply chain issues, not every customer is the most desirable. I mean, I'm sure you go through a triage internally about how to evaluate what's going to work. And so when you look at potential, the patient at the end of the process, how do you then work with the customer in order to optimize both your time and the partner's time?
That's an interesting question, Chris. So from our perspective, we are keenly aware that these molecules are our customers, their babies, right? So many of these molecules are going to address unmet needs. They are going to be best in class, first in class. So we are very, very aware that being reliable from the customer perspective is very important, but also important to understand the customer's particular need. Is it timeline? Is it the quality, let's say for a biosimilar, quality of the product? Is it that they are also still going through fundraising and therefore there might be some pauses in the development steps, et cetera. But we consider all customers to be our key customers, essentially, small and big, and be able to meet the customer where their needs are. Yep.
So have the hard conversations at the beginning and hopefully no more hard conversations.
Absolutely. That has been a learning through this 30-year journey in terms of very upfront discussion, especially for early phase molecules where there might be development challenges, being able to, we see ourselves as consultants, right? That we give the customer a truly unfiltered sort of information and sort of projection how difficult this journey might be for them. And if we think the molecule is not ready, we'll tell them that as well.
Yes, I started out this year saying that it's going to be the year of more partnerships and collaborations, and many groups are realizing that it's not a transactional activity, it's really
That's right.
yeah. So keeping the focus on the patient for a little bit, could you talk about the CLD program, which seems to be a rebate program, and why did you launch it?
Yeah, so CLD, cell line development, launching of this rebate is all about our focus, again, to get molecules into clinic to prove their efficacy. And basically, this program allows us to help our customers, to those who potentially have some funding challenges, to be able to move fast from early development stage into clinic. And we feel this goes with our mission to move into the clinic. What we especially see with the current funding environment, that for a lot of clients, showing that clinical data is important for them to get the next level of funding, right? That always didn't used to be the case, but definitely the expectations from investors have gone up, right, in terms of clinical data. And that's where we want to be supportive of our clients, essentially.
Well, just in the brief time we've spoken, it's obviously you know this field very well. So talk about the financial incentive. There's a sort of a 500K element to this program.
Right. So the incentive is basically the cell line development from gene to get to research cell bank is about 500K, which is probably one of the lower end of service cost you'd see for cell line development. But we are essentially making it free by saying that if you're signing from cell line to GMP, first GMP run, you'll get a $500,000 rebate, essentially. So you'll have to spend, about $500,000 less than you were planning, and that comes at the point when you are doing the GMP run.
Yeah, so that's a lower barrier to entry for a certain class of. So I would like to ask a sort of a closing question, but before I do that, you interested me with the origin story and the science, and particularly the analytical technologies. What technologies have you seen that have been promising and helpful, and what would you most like to see in your daily work? If you could solve a technology problem, just one, what would it be? So
one of our pride is our analytical capabilities, not just in terms of the depth we have, but the breadth. So we have, for example, mass spec center of excellence. We have cell-based assays center of excellence, particle analysis for drug products. A lot of customers, a lot of CDMOs reach out to us as well. So for all of us, though, in the industry, we want to know if there is a failure mode earlier than later, right? In terms of the molecules, activity, and potential performance in the clinic. So in terms of a particular assay, I honestly think everything that's available in the market we have, but what I see with AI coming into play, I see a lot of potential for early assessment of molecules characteristics, right? So that you can predict if there would be process development challenges later on. So that is something what we are investing and focusing on now.
Well, actually, I was going to sneak that in as well. Like, because of AI, are you finding it a rich mine of data and, I guess, strategy when you look back at past programs and what you've learned to avoid?
So we believe it has that potential. I think others are seeing that potential as well. We also see the challenge of not all of our data is in a digital format. So we are going back and structuring all the historical data in a way that we can easily harness the data and look for those similarities, right? And just at a protein structure level, be able to say what kind of challenges we would have. We are seeing early potential from it, but the work has just begun. It's a huge volume of data.
Yeah, it's a bridge that still needs to be crossed for a lot of people. So wrapping up, what does the CLD rebate program say about your company and who it is at its core?
What it says is that we are committed to get our clients to the clinic. We are committed to help them at a critical stage where a lot of clients are having to pause because not having funding. So being able to take from cell line to the early stage tox study all the way to clinic, it goes with our DNA, which is really we see our success if the molecule goes to clinic and molecule moves through all the way to market, right? As opposed to did we stay within scope and did we do our little part of the work? So that ethos matches with this rebate essentially given the current market environment.
Yeah, so you're trying to mirror as much as you can the need with the customer and the patient.
Exactly.
Yeah. So any questions for Sigma and for KBI? So I'll ask one last question, which is which activity are you seeing the most traction in recently? I mean, Bio a couple of years ago was here in San Diego and ADCs were the big news. Are you seeing more interest in, this is going to sound like a weird question, like in vaccines or mRNA?
I think really more interest in more of the conjugates, I'd say, right? Not just antibody drug conjugates, but other types of siRNA and all kinds of diversity.
Peptides?
Peptides as well, absolutely. Yeah, we are getting into peptides as well in terms of biologics produced in E. coli. Yep.
Well, thank you, Sigmund. It's been a great
Thank you,
pleasure.
Chris. Thank you.
Thank you.